When a man makes post-copulatory investment in the woman and her offspring, as humans typically do, he must be sure that the offspring is indeed his. This entails mate guarding to ensure her fidelity and avoid cuckoldry. Continuous sex had the added bonus of holding a man's attention and cementing the pair bond. Recent research reveals that both oxytocin, a neuropeptide associated with increased trust and decreased fear, and dopamine, a neurotransmitter associated with desire and reward, are triggered simultaneously by copulation in monogamous species. THIR co-action is thought to underlie partner preference and pair bonding. Young and Wong. 2004. So, according to this scenario, women evolved concealed estrus to keep their male partners around. But the uncertainty of paternity introduced by concealed ovulation had another and rather opposite effect. It allowed women to take multiple mates and, because each man believed he was the father of her offspring, he would act paternally toward it. Hurdy, 2000. The original impetus to this hypothesis was Sarah Hurdy's 1979 discovery of the high rate of infanticide in langur monkeys that often accompanied the takeover of the group by younger, incumbent males. Infanticide effectively cleared out the genetic legacy of the previous males, wiping the slate clean to begin again. The costs to females were extreme. Perhaps then, women used concealed estrus and continuous sexual receptivity not to capture one man's interest but to ensure many men's paternal tolerance. Among the Ake of Paraguay, 3 types of fathers are recognized: the man to whom the pregnant woman is married, the man with whom she had sex just prior to her pregnancy, and the man whom she believes is responsible for the pregnancy. About 63% of babies have multiple fathers or partible paternity, and the average number of fathers is just over 2. Because the Ake sometimes dispose of a child whose father dies, children with multiple fathers have an important advantage. Among the Bar of Venezuela and Colombia, a woman who claims multiple fathers for her unborn child runs a lower risk of miscarriage and of perinatal death, presumably because all the fathers ensure that she is well supplied with food throughout the pregnancy. Each child also has a greater chance of surviving until the age of 15 because it has the support of 2 fathers. THSA unusual cases notwithstanding, not everyone accepts the partible paternity hypothesis. Its plausibility depends on exactly how strong human paternal certainty must be to ensure investment in the offspring and on whether that investment is graduated by the degree of paternal confidence. If a man's certainty of paternity has to be high before he will invest, and men's acute sensitivity to women's infidelity and the frightening strength of their jealousy suggests this is likely, then trying to convince multiple men that they may be the child's father ought to be doomed to failure because each of them would. Women as choosy investors. 55. Have sufficient doubt to prevent their investment. THs would argue strongly for the mate-at-home hypothesis of concealed ovulation. But it may be that, under some ecological conditions, a modicum of care from several men outweighs full investment from one. THs depends upon paternal investment being graduated according to certainty rather than being an all or none affair. Men who harbor a hope of their possible paternity will make some efforts to protect the child, as long as the cost is not too great. Even if they simply refrain from harming it, the child's survival chances may be enhanced. Diamond 1997 suggests that both hypotheses are correct, but they apply to different points in our evolutionary history. By examining the family tree from which humans descended, Sillen Tulberg and Mahler, 1993, it is possible to find at least 20 points at which changes in ovulatory advertisement occurred. If we look at those species which shifted from advertised estrus to concealed ovulation, we find that the vast majority were promiscuous or harem-structured, where a newly established alpha male might well pose a threat to infants. THs seems to suggest that concealed ovulation developed originally to confuse paternity rather than to enhance monogamy. But we can turn the question around and ask what the prevailing estrus conditions were in those species that went on to develop monogamy. Monogamy never evolved in species with advertised ovulation and was largely confined to those species that had already developed concealed ovulation. THs suggests that concealment was a female strategy to retain the attention of a single male. If we put the pieces of the puzzle together, the primate data suggest that concealed ovulation arose in promiscuous or harem species in order to confuse paternity and prevent infanticide. Once it had evolved, the species switched to a monogamous mating system in which concealed ovulation worked to keep a single male at home. Given the flexibility of human sexual strategies and our ability to fine-tune our behavior to different circumstances, it may be that women exploited these two benefits under different circumstances, depending on the quantity and quality of available men. A third possibility, championed by T.H. Ornhill and Gangestad 2008, is that concealed fertility allowed women to simultaneously have their cake and eat it. A good strategy for women might be to find a long-term provisioning mate, while at the same time engaging in extra-pair copulations with more genetically desirable partners. Given that the strategy depends on secrecy, it is difficult to get reliable data from women themselves. Contemporary non-paternity rates vary between 1% in AFFLU in Switzerland, Sass, Muller, Chakraborty, and Ott, 1994, and 20% in impoverished neighborhoods, Cerda Flores, Barton, Marty Gonzalez, Rivas, and Chakraborty, 1999. According to T.H. Ornhill and Gangestad, women's mate preferences change in a predictable way around mid-cycle when they are most fertile. THEY prefer men who exhibit more masculinity. 56. Mothers matter most. Women and parental investment. In their face, body, voice, and scent. THEY prefer men with greater bilateral symmetry, indicative of high-quality genes, and efficient immune system, and developmental stability. Women who are partnered with men of low genetic quality show a more marked estrous preference for these good gene qualities and feel less committed to their partner at that time. Married women are more likely to be unfaithful at this time in their cycle and are less likely to use contraception when they are with a lover. Baker, 1996. TH's argument remains controversial. T.H. Ornhill and Gangestad define estrus in terms of a woman's enhanced but selective sexual interest when she is most likely to conceive, and maintain that women's estrus is not concealed from themselves, p. 275. But the evidence of increased sexual desire during ovulation, as the authors note, is far from supportive. If women's sexual desire is not enhanced, what motivates them to actively seek extra-pair partners at this time? If the illicit garnering of high-quality male genes is the aim of concealed ovulation, then why are women's extra-pair copulations relatively rare? After all, by definition, only one woman in the world is mated with the optimal man. Despite uncertainty about the origins of concealed ovulation, one thing we can be sure of is that it did not work to men's advantage, it evolved to benefit women and their offspring. When it comes to the act of intercourse itself, women do their best to make life, at least the creation of it, difficult for a man. More demanding and tortuous the achievement of fertilization, the greater the chance that only the best sperm will succeed. Let us view this Olympian feat from a sperm's perspective. Before our sperm can even enter the arena of the female reproductive tract, he has journeyed from the testes into the epididymis where he sits for 2 or 3 weeks before he is capable of self-propulsion. When his turn comes, he enters the vas deferens awaiting the orgasmic contractions that will shoot him from the scrotum via the penis into the woman's vagina, traveling in the seminal fluid that has been collected at the prostate gland. He does not travel alone. A man releases between 100 and 300 million sperm in each ejaculate. Sounds like a lot, but as many as 40% are defective or fail to swim properly. Vagina's usual acidity, designed to control bacteria, is thoughtfully relaxed a little as ovulation approaches. Mucus, normally impenetrable, becomes thinner and forms channels like swimming lanes to guide the sperm upward. Sperm seem to be attracted to the heat of the upper fallopian tubes and also by chemical signals. Why does it take so many sperm to fertilize an egg? Because they won't ask for directions. Thanks to signals from their female host, maybe they don't need to. But the journey is slow. Although they must travel just 10 centimeters, their swimming speed average is only 3 millimeters per minute. It is also tortuous. The sperm have to avoid the crevices and blind alleys of the reproductive tract, an attack by the woman's white blood cells. After 10 minutes. Women as choosy investors 57. Or so, some of the FIRST sperm have passed through the cervix and entered the uterus, with their slower companions arriving there in the next 72 hours. Only 1% of the original band will have made it this far. Now, hyperactivated and able to fertilize after their immersion in their host's body, a mere 1,000 remaining sperm swim into the fallopian tubes pushed forward by uterine contractions. About 100 of the strongest sperm reach the egg and use enzymes to digest the cell's outer membrane. But only one of them can penetrate the oocyte. When that FIRST heroic sperm enters, the tail which has served it so well is left outside. Newly formed zygote slams the door shut by forming a thick wall to keep the slower sperm out. Fertilization finally takes place. A woman's body subjects the hapless sperm to such an obstacle course as a form of quality control. A woman does not want to share her costly egg, loaded with expensive nutrients, with just any old sperm. A man's sperm are pitted against one another in a race to find the fastest, strongest, most active, and most viable. A whole area of evolutionary biology is devoted to the study of sperm competition. Burkhead, 2010, Parker, 1970, Shackelford and Getz, 2007. In species where females mate with multiple males, sperm competition takes place between rival males. In these species, females have lengthened their oviducts to make the sperm's trip more hazardous and so impose stronger quality criteria. Anderson, Dixon, and Dixon, 2006. It is a form of sexual selection that operates after, rather than before, mating. Male intrasexual competition moves from direct combat and extravagant display in the world outside, to tests of sperm speed and strength inside the female body. In many species, males go to considerable lengths to ensure that theirs are the only sperm in the competition, at a cost to the female. In seed beetles, for example, the male has a large barbed penis that tears the female tract during mating. The pain deters the female from remating with any rival male who might fertilize her eggs. The damsel FLY's hooked and horned penis is designed to remove the sperm of previous male suitors, and it seems to be a useful adaptation. If a female mates with two males, it is the second male who fertilizes 90% of her eggs. A similar function may explain the structure of the human penis. The coronal ridge removes an estimated 90% of vaginal contents, as opposed to only 35% in an unridged penis. Gallup and Birch, 2004. The spiny penis of chimpanzees is not found in humans, for which women everywhere give thanks. Its absence has been traced, serendipitously, to a segment of non-coding DNA near the male's androgen receptor gene. McLean et al., 2011. Monogamy, it seems, eased competition between men with happy consequences for their penis structure and their partner's comfort. Sperm competition does not mean that a female is a passive spectator to male attempts to exploit her reproductive facilities. A key lesson in evolutionary 58. Mothers matter most. Women and parental investment. Theory is that for every gambit there is an equal and opposite gambit, the coevolutionary game. Just as predator and prey are locked in an arms race, so are males and females. We'll return to this in Chapter 8. We can see this best in the avian kingdom. Among wildfowl, low-status males occasionally bully females into unwanted. She retaliates by ejecting the off-ending sperm before the upstart male has even dismounted. She rarely does this after a tryst with the desirable dominant male. Pizzery and Burkhead, 2000. And the females do not have to depend on visual recognition of the offender, the sperm alone provide the information they need. When a female wildfowl is inseminated with a cocktail of semen from two males, one more dominant than the other, she disproportionately ejects the sperm of the subordinate. Burkhead, Chaline, Biggins, Burke, and Pizzery, 2004. In humans too, about 35% of sperm is lost from the vagina within 30 minutes of intercourse. THs is what biologists rather indelicately term, FLOW back. As yet we do not know whether women, like birds, can bias their retention of sperm from different partners through this form of cryptic choice. If they can, it may be linked to orgasm timing. Baker and Bellis, 1993. If a woman experiences a copulatory orgasm 1 minute before and up to 45 minutes after her partner ejaculates, she retains between 50 and 90% of his sperm. If the woman does not climax at all or does so more than 1 minute before her partner, she retains 0 to 50%. During orgasm, in fact shortly after she psychologically experiences it, her cervix gapes open, widening the mucus channels, dipping down into the seminal fluid and allowing more sperm to pass through. The contraction of uterine and vaginal muscles creates a pressure change which sucks the semen more effectively through her cervix. The orgasm also expels old sperm from the uterus, creating space for new sperm to enter. Women are more likely to experience orgasm with an attractive rather than an unattractive partner. Shackelford et al., 2000, Ornhill, Gangestad, and Comer, 1995, and with a lover rather than a husband. Bellis and Baker, 1990. Same seems to be true of female Japanese macaques who orgasm more frequently with dominant males. Troisi and Kurosi, 1998. So orgasm may act as a mechanism that enhances the probability of a pregnancy from an active intercourse. But this view remains controversial, and female orgasm may have no functional value at all. Perhaps it is simply a byproduct of the fact that male and female genitals develop from the same underlying structure. See Barish and Lipton, 2009, Gould, 1992b, Lloyd, 2005. The presence of testosterone turns the embryo's genital tubercle into a penis, otherwise it becomes a clitoris. Some argue that orgasm is important for male sexual excitement and fertilization, but it is just an added but unnecessary. Women as choosy investors. 59. Bonus, for females. After all, females can be impregnated without experiencing orgasm at all. E versus ENA featuring sexual intercourse, women have ways of controlling whether or not conception will occur. Failure to conceive can result from a shortening of the normal 14-day interval between ovulation and menstruation, which reduces the chance of a fertilized egg implanting. It is in this time window that a number of embryos are ejected without the woman's awareness. Only about 60% of fertilized eggs succeed in implanting themselves in the uterus. Of these, a further 60% will not survive to the 12th day of pregnancy, and the woman experiences what she may believe to be an unusually heavy period. Of those that make it through the 12-day barrier, about 20% will be miscarried in the first trimester. Baker, 1996. The majority of these pregnancy failures are a result of genetic abnormality in the embryo or highly stressful maternal events. Miscarriage rates increase with the outbreak of war or with the death or infidelity of a woman's partner. Even when a pregnancy is established, women still exercise choice about its continuation and even about whether to care for a child after it is born. If a fetus or infant is not viable, then a woman is better off abandoning it as early as possible. Trivers, 1972, argued that this was because the longer she continued to invest in a child, the more time and effort she had wasted when it ultimately died. As was christened the Concord fallacy by Richard Dawkins and Tamsin Carlyle, 1976. EY drew an analogy with Britain's sustained and economically disastrous commitment to the supersonic airliner, which was driven by the logic that so much had already been spent that it was unthinkable to abandon it. Dawkins and Carlyle pointed out that this retrospective view of investment to date, however intuitively appealing, was incorrect. EY argued, and Trivers, 1985, later came to agree with them, that the true explanation of early abandonment lay in the amount of prospective investment that remained. A woman who abandons a non-viable fetus saves herself 15 years of future wasted investment, while a woman who abandons a 12-year-old saves herself only a further 3 years of investment. THSA interpretive arguments notwithstanding, the evolutionary predictions are identical and are not in dispute. Cruel truth is that from the point of view of her own genetic success, a woman who spends time and effort in raising a child who is a poor reproductive prospect or who will be born into an environment too harsh to sustain normal development curtails her chance of investing in other more viable offspring. Natural selection should favor female bodies and minds that are profoundly selective about which infants deserve the mother's scarce and valuable investment. In many contemporary cultures, women have the option of terminating the pregnancy. We would expect that this decision would be related to whether our 60. Mothers matter most. Women and parental investment. Not the mother would do better to wait for a more propitious time to bear the child and to the currently available resources for rearing the child. Decision to have a child now, rather than waiting for better emotional and material circumstances, is closely related to the mother's age. Younger women have a longer reproductive career ahead of them and can afford to wait for a more optimal time. Statistics confirm that the rate of voluntary abortion is high among young women aged 20 or less, with approximately 3/4 of 18 to 19-year-olds choosing to terminate their pregnancy. Hill and Lowe, 1992. Young women can afford to wait not only in terms of their biological clocks but because they have a better chance of finding a male partner to assist in child-rearing in the future. Younger women have a much higher probability of marriage and remarriage than do older women, and childlessness increases their likelihood of finding a marriage partner. Material, personal, and interpersonal resources also play a role in the decision to abort. Fathers make a contribution to the economic and emotional welfare of the mother and child, and statistics confirm that the best predictor of abortion is whether the father is willing to commit himself to the mother. In the United States, 65% of pregnancies among unmarried women were terminated compared to 10% among married women. Hill and Lowe, 1992. A study of 300 middle-class American women asked them to estimate the probability of terminating a pregnancy. Essock, Vitale and McGuire, 1988. The estimated probability was 0.07 for married women but 0.64 for unmarried women. Fathers are less likely to contribute to the care of a child who is not their own, and, when married women were asked about the likelihood of abortion given uncertainty about who the father was, the probability rose to 0.57 in line with the figure for no supporting male. Fathers are not the only source of possible support. State aid may be meager, but it is better than nothing. In a study of teenage girls who made different choices about their pregnancy, the provision of state aid decreased the abortion rate to 42% compared to 71% among those without such aid. Leibowitz, Eisen, and Chow, 1985. A woman's own psychological resources are important too. Women suffering from psychological instability may lack the social, emotional, and physical resources not only to rear a child but also to retain a long-term male partner. In a comparison of women suffering from anxiety and depression with a non-clinical control group, the clinical group was less likely to have ever married, 70 versus 94%, and far more likely to have divorced, 87 versus 38%. THSA women had a 0.39 probability of abortion compared to 0.07 for the control women, although the two groups did not differ in their likelihood of ever having been pregnant or in the probability of miscarriage or stillbirth. Essock and McGuire, 1989. Women as choosy investors. 61. Infanticide is an act that not only excites feelings of revulsion but one which at first sight seems wholly incompatible with evolutionary theory. Yet given the extreme time and resource commitment demanded of human mothers, we should expect infanticide to be a line of last resort when circumstances militate against continued investment. Although abortion has effectively removed the need for infanticide in many societies, in others it may be the only avenue for a woman who bears a child that she cannot support without severely compromising her existing children. In the United States, infanticide is mainly confined to young women who effectively deceive themselves and others into believing that they are not pregnant at all. After giving birth alone and in secret, the young woman disposes of the child and may even convince herself that she has done nothing wrong. Some believe that the child belongs to them, and as such they have a right to decide upon its life or death without state interference. In other cases, the woman convinces herself that the child was stillborn and that she has not committed any offense. Pearson, 1998. From the point of view of evolutionary theory, we would expect to see more frequent infanticide when the infant's health is so poor as to cause doubt that it can survive to reproductive age. Daly and Wilson, 1988, consulted cross-cultural evidence from 60 societies around the world, noting in each case the reasons that were given to explain the use of infanticide. 21 societies indicated that deformity at birth or serious perinatal illness would be grounds for infanticide. 6 societies recognized infanticide as a response to the absence of male support for the child and economic hardship. Twins place an extra burden on the mother and on the available economic resources. Usually one rather than both of the twins are sacrificed. And infanticide is less likely in societies where the mother can give up arduous work because she has female relatives or friends to help her. An infant that is born too soon after a previous birth is also more likely to be killed. Mother must decide between the older child's need for her milk and that of the new arrival. In line with evolutionary predictions, the favored child is the one in whom she has already invested most. If the child is the product of a clandestine extramarital affair, the chances of infanticide rise in line with the potential threat that the infant poses to the continued provisioning of older children by her husband. Contemporary data from Canada and France. Daly and Wilson, 1988, support the idea that infanticide is related to the mother's evaluation of her current economic and social situation as well as her future likelihood of giving birth. From the woman's reproductive condition before the act of intercourse to her emotional attachment of her newly born infant, there are many points at which a less than optimal conception can be unconsciously or knowingly prevented or halted. Once the baby is accepted, the woman commits herself to nearly 62. Mothers matter most. Women and parental investment. Decades of care, protection, and concern, small wonder that such an investment is carefully made. Women as heavy investors. Those mothers who were good carers and protectors had more surviving offspring, and psychologists are beginning to understand how evolution has crafted a maternal brain. As knowledge has come from studies measuring changes in the key neuromodulator oxytocin during pregnancy and beyond, and from imaging studies which show graphically the changes in the mother's brain that occur in the weeks after giving birth. Oxytocin has received a lot of media attention and become popularly known as the trust hormone. Some enterprising, if misguided, companies have even tried to market it as a kind of scent. Despite its sudden rise to prominence, versions of oxytocin have been around for 700 million years and its homologs are present even in insects. How its precise function varies across species, oxytocin in mammals has long been recognized as important in the birth process where it triggers the smooth muscle uterine contractions needed for delivery. Doctors use a synthetic version, pitocin, to induce labor. It is also involved in the milk let-down reflex necessary for breastfeeding. But oxytocin has psychological as well as bodily effects. Bodily effects occur because it acts as a hormone traveling through the bloodstream to reach many parts of the body. After being synthesized in the hypothalamus, the magnocellular neurons of the paraventricular and supraoptic nuclei, it travels to the pituitary gland where it is released into peripheral blood circulation. But oxytocin has another route taking it to many parts of the brain. Starts in a different part of the hypothalamus, the parvocellular neurons of the paraventricular nuclei, which projects to ancient, old brain, limbic sites hippocampus, amygdala, striatum, hypothalamus, nucleus accumbens and to the brain stem. In short, oxytocin looks like a good candidate for orchestrating mothering at a psychological as well as systemic level. Early research that pursued this idea was on rodents, see Broad, Curley, and Kevern, 2006, Insel, 2000, Kendrick, 2000. In pregnancy, triggered by rising estrogen levels, oxytocin receptors are upregulated in the brain as well as the uterus. Vaginal stimulation of birth activates the hypothalamus to release oxytocin to many brain areas responsible for maternal behaviors such as nest building, pup retrieval, licking, crouching, and maternal aggression. A stunning experiment demonstrated just how powerfully oxytocin could affect mothering. Peterson and Prange, 1979. When oxytocin was infused directly into the brains of virgin rats who normally give pups a wide berth, they began to women as heavy investors 63. Behave like mothers. And it worked the other way around too. If an oxytocin antagonist, a neurotransmitter that blocks oxytocin's access to its receptors and therefore renders it ineffective, is given to a new mother, it prevents the normal onset of maternal behavior. Insel, 2000. In humans too, prenatal and postpartum oxytocin appears to enhance the mother's initial bond with her infant and reduce her stress levels. Newman, 2008. We cannot see the chemicals inside a human brain, but we can use spinal taps to measure the contents of the cerebrospinal fluid in which the brain is bathed. During childbirth, these central levels of oxytocin rise and postpartum plasma levels are correlated with positive feelings and reduced anxiety. Takagi, Tanazawa, Itsuki, Haruta, and Yamaji, 1985. Immediately after birth and prior to their FIRST feeding, infants massage the mother's breast, causing oxytocin levels in her blood to rise and remain high during feeding. Mathieson, Rancho Arvidsson, Nissen, and Uvnes Moberg, 2001. Oxytocin lowers maternal stress and increases feelings of calmness. According to women's own reports, breastfeeding lowers subjective stress and negative mood states. Mezzacappa and Katkin, 2002. Is as evident at a biological level too. Lactation is associated with lowered activity in the stress response system, the hypothalamic pituitary axis, visible in lower levels of cortisol and other stress hormones. Amico, Johnston, and Vanucci, 1994. Recently, researchers have begun to examine the emotional and behavioral effects of oxytocin. In one study, researchers tracked oxytocin levels in women's blood during the FIRST and third trimester of pregnancy, and then during the FIRST postpartum month. A stronger attachment between the mother-to-be and her fetus was associated with a pattern of increasing oxytocin during pregnancy. Levine, Zagori, Sharon, Feldman, and Weller, 2007.